first, again, I told you, yesterday our reference was flipping card. Just in some cases, other references I was used. But today, completely are teaching you based on flipping card by Mr. Book. If you don't have the PDF file of this book, I can share it with you. Okay? If you want, you can inform. Guys, at first, I will say you an introduction. After that, we will work on nomenclature and classification of enzymes. After that, we will speak about properties that you are familiar with all of these things, but we want to expand the subject just. The mechanism of enzyme actions and enzyme kinetics and enzyme regulation and the role of enzymes in clinical diagnosis is the subject that we will discuss. At first, guys, as you mentioned, enzymes catalyze chemical reaction. Are they consuming during catalyzing the reactions? No. They are just speed up the reaction, but they won't consume. This is the thing that I told you, and they have no change overall the process. If there was some changes after the process, that will be again regenerate and become reversed. The enzyme have protein base generally, but we have some exception. Which of them? All of the enzymes is made up of proteins. Do we have any exception?
Yes, there are. Steroids?
No, not steroids. We have an enzyme under the name ribosine. Ribosine or ribozyme. Ribozyme is made from RNA. They are in our saliva and at first have antibacterial effects, and also they have some roles in protein synthesis, a little in some parts, for producing peptide bonds. Okay? And in another situation, I want to tell you that the mechanism of some drugs are based on enzyme inhibition. That I want to explain you completely. On the other hand, guys, enzymes are related to the human well-being. Imagine, if a baby comes and have some deficiency in urea cycle enzymes, just one enzyme, does that baby have normal life? No. The deficiency of enzyme have critical role in some situations, like some inborn error of metabolism. That's just one single enzyme can't work properly. And they have role in well-being. On the other hand, guys, the enzymes can be used in diagnosis. Can you tell me how? Diagnosis of some diseases.
Genetic disease?
We can, but the evaluation of the genetic disorder, it's not easy. There is some molecular techniques. What more easier? Yesterday I was mentioning about two liver enzymes. Am I right? ALT, AST. They are enzymes that we can use for evaluation of liver function. Okay? So, they can use in medical situations in different areas. Another thing, they have role in industry for producing some food. For example, for producing the cheese from the milk. We use rennin. Rennin is an enzyme. You will work on that an enzyme practical session. And in different situations, for example, in detergents, for example, some soaps that we are working or some material that we are using them for washing clothes, they are full of enzyme. You can see that they are written on that they are made up of blah blah enzyme. And on the other hand, they can use them for increasing nutrition value and healthy made foods more supplemented somehow. And I told you that they are used for diagnosis and prognosis, and that I will explain you more. Guys, what do you know about nomenclature and classification of enzymes? What do you know?
The suffix is used for enzymes.
Yeah, the suffix "ase" is generally used. The "ase" can add to the substrate name. Am I right? For example, we have an enzyme that converts urea to CO2 plus H2O. We call it urease. The "ase" suffix is added to the substrate name. Maybe they add the suffix to the reaction name. Like lactate dehydrogenase that converts lactate to pyruvate. The dehydrogenation of lactate is catalyzed by this enzyme. And in some cases, they should be the name of some trivial name and some old name of the enzyme, like trypsin, like chymotrypsin, like pepsin. They are not based on a special reaction, but they are based on the old name. You can see here a substrate name plus amylase, lipase, urease, and on the other hand, lactate dehydrogenase, adenylyl cyclase. Do you know the role of adenylyl cyclase? What does cyclase mean?
To convert the branch of single chain structure into cycle conform.
Yeah, exactly. Adenylyl cyclase that convert ATP to cyclic AMP. This is adenylyl cyclase. You will study completely in hormone and some endocrine blood. And about the enzymes that there is no relationship with reaction or substrate name, trypsin and pepsin. Do you know about these enzymes? Where are they working? Stomach and digestive tract. Okay? This was about the name and the nomenclature of enzymes that you were familiar with them. Am I right? No? I am not right. This is the first time that you've heard about these names. Okay. But they are more common. But we have
Can you repeat the last one? Can you repeat the last one?
about pepsin and trypsin.
Yeah.
Those enzymes, they don't obey a special rule for naming. Yeah? There are just old names, for example, trypsin and pepsin, that they are enzymes in GI or gastrointestinal tract, especially in the stomach. We have them. Okay, They are proteins. They're breaking down the protein structure. Okay, that was about the recommended and general name. But we have another systemic name system that they are a little, not a little, they are complicated, more complicated than usual, and we don't use those terms in general. They are based on this description of chemical reaction and all the substrate plus suffix "ase". You can see here, instead of LDH, the systematic name is lactate, nicotinamide, aldehyde, dinucleotide, and plus oxidoreductase. You can see the complete name of substrate, co-substrate, cofactor, and the reaction. And another classification that is based on enzyme consensus, that we have some codes, that it is not important to memorize these two items. Just bring them, that if you see here, this is the LDH. Guys, do you know what is the benefit of this naming system?
Makes it easy.
Easy for scientists. They decrease the ambiguity. It is clear if you type even in Google EC 1.1.1.27, it will raise for you the evidence. Okay? That was about the schematic classification. Right. If I want to tell you about 6 major enzyme classes, you can see here oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. Guys, you should memorize these categorizations. You should memorize all of these 6 classes in order. Okay? It is necessary because you have, I'm sure that you have in your basic science exam, require exam 2. In every entrance exam, we will working on them. Okay? Do you have another exam from BS to MD in Iran?
Any for MBBS.
Every BS, newly every BS. From bachelor degree, science composition.
What if you study from MD? What if you start from MD?
Start from MD, no. After they go, become graduated from bachelor degree, they have an entrance exam and come for MD, not from the first. I was searching for questions of that exam. I can see this exam classification on those exam even. This is so clinical exam. It's not just basic exam, but I can see this classification. Okay, this is the 4 classification that we will speak about them one by one. Guys, again, we have here lactate dehydrogenase. What do you think about the name of the enzyme, the word of that they are doing?
Both oxidation reaction.
Exactly. They do oxidation and reduction. With adding oxygen or removing electron or transferring H+, all of them categorized in oxidoreductase system. That they catalyze oxidation and reduction. You can see here that electron and proton is transporting. It's not, guys, important to memorizing the reaction, just about the enzyme class. Okay? If I ask you, for example, the lactate dehydrogenase is related to which class, you should tell me oxidoreductase class 1. This was about oxidoreductase. Another class, the second class, is about transferase. What are they doing? Transfer a specific group. For example, here C, A, or P containing groups. And about guys, phosphorylase that transfer phosphate that we have in enzyme, it is related to this class. Or ALT, alanine aminotransferase. They are transferring amine group. Okay, these are in transferase group. And the third class is hydrolysis. What are they doing? Adding water and OH and H for breaking down one molecule. You can see here urease is in the classification of hydrolysis. This was related to the third class. And about the fourth class, this is liases. This is a little, I think, difficult, but memorize that all the carboxylase enzymes in the categorization of liase. What are they doing? They cleave the bonds between CC, carbon-carbon, carbon-sulfur, and some carbon-nitrogen bonds that you can see here. And release, in some cases, CO2 or H2O. They cleave the CC bond. This is another class of enzyme. Guys, remind me, after telling the last class, tell you about these two enzymes and the I will explain what I just came here for memorizing. Guys, the next class is isomerase. What are they doing? They convert different isomers to each other. Am I right? For example, here you can see that methylmalonyl-CoA is converted to succinyl-CoA and all other isomers that we can see. Another thing is about ligase. What are they doing? They actually ligate or attach another thing to another molecule. Here you can see that pyruvate carboxylase attach CO2 to the pyruvate. And guys, be careful. This reaction needs energy in the form of ATP. This is so important. You should know that these enzyme class need ATP for doing the reaction. And about synthase and synthetase. You will face with these two terms in your metabolism block. I think in the next semester, inshallah. Guys, synthase, salam alaikum. Synthase need, at first, they are in liase classification and need no ATP. Need no ATP. But synthetase is related to ligase and need ATP. Be careful that this synthase and synthetase is not some typos rhyme. These are different classes of enzymes with different forms. These were about just enzyme classification that we had 6 classes that we should know about the work that they can do, but not necessary to memorizing the reaction class. But be careful. For example, carboxylase is in the group of ligases, or decarboxylase is in the group of liases. Okay? Be careful about them. Guys, about the enzyme properties, I want to tell you some important issues related to active site, efficient enzyme, we are speaking about generally protein enzymes, okay? Because RNA enzymes are the exception and just we have ribosome. It is not important. Enzymes generally have an area under the name of active site that is produced by conformation of protein structure. Different folding of protein in which hierarchies structure we have primary, secondary, tertiary, and quaternary. For example, imagine this is an enzyme with one subunit. This is which classification, which hierarchical structure? Tertiary. Okay? Tertiary structure that have three-dimensional shape. Guys, you can see here that this enzyme have active site that can cooperate with substrate or reactant. Both are correct, substrate or reactant. There is a cleft or pocket that can substrate attached to the enzyme and have some interaction with enzyme. For example, some hydrogen bond or something other, some hydrophobic bond, and after the reaction completed, it converts the product and again the enzyme is regenerated. There are different models that I want to tell you. At first, guys, there are two different models for mechanism of action of active site. One of them is about lock and key model. What did it say? Can you tell me?
So the enzyme acts as a lock, and the substrate acts as the key because the key will only fit in a specific lock. So then when it attaches, it's called the enzyme substrate complex.
Yeah. Did you guys get the point, or I should explain more? All of you get the point? The enzyme is a lock and the substrate is the key. This is completely fitted to the active site. But another more acceptable model is induced fit model. What did it say? What did it say?
The complementary shape of the enzyme and the protein is different, but the close connection between the enzyme and the substrate will make the active site in a shape that is complementary to the substrate.
Yeah, did you get the point? Guys, exactly, it is like a hand glove that you put your hand on that. For example, imagine this is a glove that just is here. After that, you put your hand in the glove. The shape of that is become exactly like your hand. Okay? This is like that. Here, the substrate doesn't have complementary structure for enzyme. After it become near the enzyme, some changes occur, that some interactions occur, that enzyme fit to substrate. This is induced fit model, and because of the protein structure, that it's not constant, it is more acceptable model. Even in some new books, they don't mention about this model at all, and just explain about induced fit model. Guys, about the efficacy of enzymes, enzyme catalyze the reaction so efficient. What did I mean? If I don't have enzymes in our body, nothing was possible. The enzyme increased the reaction rate 1,000 to 100 million times faster than normal. Without enzyme, the life was not possible. Okay? They speed up the reaction. Another term that I want to tell you is about turnover number, or K-cap. What did it say? The number of substrates that convert to product by one enzyme molecule per second. It is turnover number, or K-cap. Did you get the point? And guys, about the K-cap, it is a constant rate, and it is the rate of conversion about ES to EP. At first, before we continue, it is better to say sooner about these. Guys, what is this? Enzyme. What is this? Substrate. It is the enzyme substrate complex. After that we have EP complex. After that we have E plus EP. Okay. The K-cat is related to ES to E plus P. This was about K-cat. That we will work on this equation after I will prepare this for 1 hour. Guys, about specificity, I think our friend mentioned about that, about specificity. What do you say about the specificity of enzyme?
A particular reaction organized by enzymes having substances.
Yeah. And one enzyme just is made for one substrate or limited substrate in some cases. Okay? And they are so specific. As our friend mentioned about that. And another point, guys, the enzymes that are in specific cells tell us and determine that which reactions occurs in that cell. Why, for example, we are saying that urea cycle occurs in liver?
Because the enzymes
Yeah. Why we are saying, for example, the glycolysis is occurring on the cytosol? Yeah. Or why we are saying that, for example, beta oxidation doesn't occur in the RBCs? Why? Do you know beta oxidation, burning of fatty acids?
because the enzyme catalyzes the reactions.
Do you know why, guys? The fatty acid oxidation, beta oxidation, occurs in mitochondria. Because RBCs have no mitochondria and the beta oxidation won't together. You can see that the organelle and the cells are specific for each enzyme is specific for different organelles or different cells. Guys, do you know about coenzyme, ecoenzyme, cofactors, and coenzymes? You don't remember. Guys, at first, the enzyme is a protein that naturally it is inactive. We call that apoenzyme. Apoenzyme. It is just protein, part of the enzyme. That is not active. After that, we add another thing that should be added there is coenzyme or cofactor. I will explain about that. In this situation, this is protein and enzyme activator. After attaching this coenzyme to the apoenzyme, now we have holoenzyme. If I ask you, I had some question for the history student on Monday, and one of the questions that I asked them for the final exam was about which of them are functional enzymes. Holoenzymes, apoenzymes, cofactor, coenzyme. Holoenzymes. Okay? This was about holoenzymes can be. Generally, guys, coenzymes, generally, in lots of cases, they are vitamins. MAD is the derivative of vitamins. Am I right? This is related to niacin. We will study about that. Coenzymes are derived from vitamins in some cases, not all cases. And if the coenzymes be related to enzyme covalently and be the functional part of the enzyme, we call that prosthetic group. Yesterday we spoke about that. What was the example about prosthetic group? Heme. Heme in hemoglobin exactly was prosthetic group, heme with iron. But co-substrate is the situation that just for working attached to enzyme and after that dissociate. Okay? Okay? Okay. Co-substrate is an organic molecule that just for working of enzyme come to enzyme structure and after that dissociate from the enzyme.
So does this activate that enzyme or
Activate the enzyme. It is necessary for enzyme function, for coming substrate, but it's not connected covalently with enzyme. And it is not in the part of enzyme structure naturally. But prosthetic group, for example, we have some enzyme under the name cytochrome oxidase. They have heme. The heme is the prosthetic group of them. Okay, this was the differences between prosthetic group and co-substrate. That both of them are coenzymes. But metal ions, we call them just cofactor. Cofactor can categorize to metal ions and coenzymes. If it was metal ions, we name them as just cofactor. But if it was other thing except metal ions, we call that coenzyme. Did you get? No? Yes? No? What should I do? Do you want I tell you again? Let me read something to me. This is our enzyme. Okay? For working, it needs something that we call that cofactor. This cofactor can be some metal ions. For example, maybe it be calcium that we have in clot activating in system. Do you know about clot activation and something of clot formation? Yeah, those enzymes need calcium, for example. This is a cofactor of this enzyme. Another enzyme, for example, needs NAD+. It is actually vitamin B3 or FAD. It is vitamin B2. Okay, these are cofactors, but because differentiate between metal ions, we call them coenzymes. Am I right? Now, these coenzymes may attach to the enzyme covalently. For example, this is your enzyme structure, and this NADH is the part of enzyme structure, not just attaching here temporarily. This is there permanently. This is part of enzyme structure. We call that prosthetic group. Did you get the point? are. These enzymes for working need NADH, but NADH is not in the structure of enzyme, just come from the environment, for example, not from cytoplasm, and NADH converts to NAD plus, and the reaction just occurs. It is co-substrate. It's not prosthetic group because it's not in part of enzyme structure. Did you get? Yeah? That was about cofactor, coenzyme, and metabolites. Guys, here we can see just some examples about coenzyme, different vitamins. For example, biotin is with vitamin B7, coenzyme A is vitamin B5. You will study, the order of lectures because of some educational changes that occur. You come here late. The order of lectures changed because we can finish that on time. And actually the prerequisition of enzyme lecture I think is vitamin. I think it is because of that you are a little confused. They are different vitamins. Vitamin B1, B2, B3, B6, B5, B7, and B12 and B9. Here you can see other metal ions that we call them as cofactors. And these are the enzyme examples that work with these cofactor and coenzymes. But it doesn't need to memorize this one. Just there are some examples. Just after. No. Finish.
Of course, why not?
Yeah, he was listening precisely after that I told him this. Okay.
Doctor, we have so much things to do.
Yeah, why not? All of us. Just we should make priority for our work in a schedule. All of us, we are so busy. And we are just lots of people that want from us, something from us. What from you is your
We
professor
learned this in high school.
in high school, yeah?
You memorize it, we don't.
It was a lot.
We memorize the function also.
Yeah, like in the
It's a
science.
bit clear.
It's not important because after a time everyone will forget if don't practically work on that. Guys, another issue that I want to tell you is about enzyme regulation. Enzyme can be regulated, activity of them can be regulated. The activity of them can be increased or decreased based on what? Okay? Please.
Like you want the factors or you want the basic, why do we eat the veggies?
Based on our cell need. Yeah? For example, we just here eat lots of pizza. The amount of lipid in our blood circulation start to increase. The digestion of that is vital. Now, the expression of some enzymes and some hormones that are related to the lipid metabolism start to increase. Or for example, today you had just carbohydrates for your breakfast. At first, the insulin level should start to increase. So based on your cellular need, your body need, the expression and the regulation of enzymes will occur. They start to become increased or decreased. Guys, another thing that is important is about the regulation and location of different enzymes within the cell. I told you about these subjects. You can see here that, for example, in mitochondria, I spoke about the specific enzymes in different organs. For example, this enzyme is related to liver. This enzyme is related to muscle. This enzyme is related to kidneys. But now it is about different enzymes that are related to different organelles. Why these are arranged in this way? For example, mitochondria have different different cycle and different enzymes. Cytosol have different pathways that we will study all of them one by one. Or nucleus have different enzymes for RNA and DNA synthesis, and lysosome again have some enzyme for degradation. What is the logical and the logical issue and the rationale behind of this arrangement? Why? Why some enzymes is in some tissue, some enzymes are in some organisms? Why?
Depends on their function. It depends on their function, not whether they can.
Because we are, as a human, as a creature, we have lots of cells. And we have millions of metabolism pathways and millions of different reactions that are occurring simultaneously in our body. If these enzymes want to work properly, they should somehow compartmentalize in different organs and tissues. Am I right?
Yes.
They should completely become compartmentalized. For example, the place of this enzyme is lysosomes. Which enzymes are concentrated in lysosomes? Hydrolase. If this hydrolase be in blood circulation freely, is it okay? No. Or maybe, for example, this enzyme be freely in the cytoplasm. Is it okay? No. So, they are compartmentalized for better function. Guys, have attendance list for a little resting. As for Fatima, we don't have. Just I will say the name that yesterday was present. After that, if someone was that I didn't say the name, tell me. Mason, Abdullah Bahar Saadi, Safi, no. Khorshid, Kolkata, you, and for molecules that want to react, guys, here we have something under the name energy barrier. Okay? The reactant or substrate should overcome this energy barrier. Should overcome. And we call it here transition state. It is the situation that we have ES. This is transition state that this substrate and enzyme should overcome this situation till can continue the reaction. It is so important, and enzyme decrease this transition state. At first, this was the thing that I explained just now, just written here for better understanding. Another thing that enzymes do, the number of molecules that have enough energy for overcoming the barrier is increased by enzyme. The number of molecules that can react increase. And the lowest energy of activation lead to have more molecules to have energy and can speed up the reaction rate. It is again another more formal form. You can see here the alteration in free energy or delta G. Do you know about delta G?
Yes, gives free energy.
Yeah, gives free energy. You can see here that initial state is the same in both situations with enzyme and without enzyme. And the enzyme kinetics that we should know. We will discuss on factor affecting reaction velocity, okay, about Michaelis-Menten kinetics, about Line-Weber plot, and about enzyme inhibition mechanism. Did you study about each of them? All of them? How? That wasn't high school, that was university.
Allah, you can check my notes. Everything is here. Everything you say is here.
Okay. What about you?
No.
It's not typical. Are you were in the same?
No, he is different country. I'm from India and he's from Sri Lanka. So different.
So neighboring countries different same.
But they bother you, no?
Yes, yes, really good.
I know
At
that.
that time,
They weren't practical for you. Why you should study these things in school time?
UK, we cannot have fights with UK.
I will say this point to my boy, to my son, because all of the time he said, in Iran everything is in bad form, we have difficult lessons, we have you've studied the things that just we start studying a specific major.
It's good. It's not even in so much detail. We have everything
Yeah.
in detail.
You should prove everything.
Yes, we have basic questions. We have everything.
Is it possible
We-
that I sit down here and you explain
Yeah.
these subjects? Oh, okay, because you are now as medical doctor, okay? The proving equation, for example, for Michaelis-Menten kinetics is not applicable for you. The clinical importance is so important, is so crucial and vital. But I think in your school, it wasn't a good situation. You can have some complaining to some area for saying these things in your school ages.
We didn't. We had no choice. Whatever they did, we did.
What was your major? Biology?
Biology, chemistry, and physics. I did two subjects.
we studied this under chemistry.
You were poorer than us. We think that just we were poor, but you were poorer than us.
No. Maths. We had maths until 10th grade. At 11th and 12th, we only did three subjects, but each subject we have six papers. So 6 and 3.
Just in 11 and 12, you had chemistry, physics, and biology, or no other things.
No.
But we had, even that, we had theory, practical, alternative to practical. So we had like 6 exams per one subject.
Okay. But just these things, not other general studies, like for example, something related to religious stuff, something related to geography, something related.
Yeah, we had. If we wanted to choose, we could choose. But I chose only 3 subjects.
For example, in Iran, we have a major under the name Ulum Tajrabi, some experimental science. Okay, we have chemistry, physics, and biology, mathematics, and beside them we have some Arabic language, English language, religious issues, Farsi language, and every other thing. What about you?
We have physics, chemistry, biology, English, and our examination is world's most difficult examination. India's examination is world's most difficult examination qualification. This is really hard.
So you guys need,
Yes.
everybody needs
Yeah.
to get in here.
But
Yeah.
for me, that's cool.
Not to get it here, but to practice
You have
there.
to get a qualified score so you can apply anywhere. But like to get in India, you have to get a degree score.
But then what you guys do need
I
is
just
work.
we have to do practice there. That is
You have hard life.
very hard.
Okay, I get the point. All of you have. So, guys, you study some of them. Some of you study some of them, and some of you, this is for the first time that you want to study. Guys, factors that affecting reaction velocity can be categorized in 4 groups: substrate concentration, enzyme concentration, temperature, and pH. Tell me, what do you know about the effect of these issues on the reaction velocity?
So in substrate,
Substrate concentration will decrease.
no.
No, it first increases and then it becomes flat. Then the concentration happens constant.
They increase and then becomes constant.
Why?
Because the active sites become
Saturated.
Exactly.
There is 2 types of substrate. There is competitive and there is non-competitive. Competitive increases the rate of the reaction, and non-competitive attacks to the electrolytic site, which causes conformational change and measures the protein and reduces the.
Do you know about these things that they are speciality?
But the
What about you?
students don't know.
I will explain. About temperature,
Kinetic energy increases, so more frequent active sites form. Oh,
And then it decreases.
why?
Because the enzymes will be natural between.
Exactly. And about pH?
Same thing. Same as temperature.
Same physics.
Somehow in some situation. I will explain. But at first, if we wanna have a definition on velocity, guys, no worry that your classmates know better than you. Just if you have any question or in some situation you don't get the point, just ask me. It is so typical and it's not a big problem because you are from different regions. Even in Iran, all of us have the same lessons, but in some situations, some students can see the point sooner than others. But asking is not a problem. Okay? The velocity is usually expressed as micromolar of product that fall per minute. It's not important to memorizing because they are confusing all of them with each other. But another thing that I want to tell you, you can see the substrate concentration, the effect of that on the reaction velocity. Here we have maximum velocity. I explained you yesterday. It was related to the which type of enzyme, and this is related to which type of enzymes?
The right one is allostatic,
Yeah.
and the left one is the normal.
Normal enzyme. Good. The normal enzymes that have hyperbolic curve, they obey Michaelis-Menten rules. Okay? They obey Michaelis-Menten rules. But about the enzyme that is allosteric enzyme, they don't obey Michaelis-Menten rules. Typically, today we are speaking about enzymes that work with Michaelis-Menten equation. But know that increasing the substrate concentration at first increase the reaction velocity till when? Till all of the active site be saturated and occupied by substrate. After that, the rate of your reaction will be constant. So, the hyperbolic curve is related to Michaelis-Menten kinetics, and the allosteric one is not related to Michaelis-Menten kinetics. Guys, do you remember that this sigmoid shape was related to what? What property of allosteric enzymes? Yesterday I told you. No?
The partial pressure of oxygen.
This was related to hemoglobin. But I told you that this effect is related to
Oxygen.
that was related to cooperative effects of the subunits of those parts, hemoglobin, and here is related to allosteric enzyme. Guys, allosteric enzymes have more than one subunit. They have active least 2 subunits. And they have cooperative effect. When one subunit attached to substrate, other conformational changes make easier to have another attachment of other cells. Another thing, all of you know about that, about the enzyme concentration. Imagine our substrate level is unlimited, is excess. If we start to increase enzyme concentration, what is happening to rate of reaction? Increase. We have unlimited substrate, and with increasing the enzyme concentration, the reaction rate is increased. About effect of temperature, guys. All of you know that protein in high temperature start to be denatured. And that's why the enzyme activity will increase to the certain temperature. After that, it will decrease because of denaturation. And this is the reason that why tell the body temperature shouldn't be more than 71, and after 40 it is dangerous. Okay? Another the effect of pH, as you've mentioned, it is again we have an optimum pH. But guys, the optimum pH is the same for all of the enzymes?
No.
How?
Some are basic, some are acidic.
Based on the enzyme reaction, I want to tell you that the pH effect on active site ionization. Am I right? They have effect on active site ionization. For example, imagine we have aspartate in active site of enzyme. Okay? And for working of this enzyme, this aspartate should be deprotonated. Now we near a high pH level that this enzyme, active site, aspartate, become deprotonated and start to boil. Did you get the point? Base, did you get? No? I can explain more. Why your eyes and your face got question marks? Imagine, this is again our ugly enzyme, okay? And in this active site, we have aspartate. Okay? Aspartate had another COOH on the side chain. Am I right? It was acidic amino acid. For working, we need that this COOH be in the form of COO minus. The pH is so important for this ionization process. And for example, this enzyme is more, for example, in alkaline situation better than acidic situation because it needs to be deprotonated. Okay? It was about the ionization. On the other hand, the pH can affect denaturation, and on the other hand, there are different optimum pH for different enzymes. You can see here that the pepsin is an enzyme on the
In the stomach.
stomach. And the pH of the stomach is near to 2, 1 and 2. And that's why the pepsin is working in that situation, but other enzymes will be inactivated in those areas. And another thing, trypsin is working in somehow in neutral pH, and alkaline phosphatase, another enzyme that work in basic pH. For example, pancreatic enzymes work in alkaline pH in our body.
still it should be basic letters. It's 7.5 to 8.
I just took the picture from a lipid cardboard, but maybe because Tristan is working based on pancreatic enzymes, it is logical that be the higher pH level, more than neutral pH, near to not 11 and not near to 8, something like this. Thank you for mentioning this. I will actually edit this part. Guys, I want to tell you about Michaelis-Menten kinetics that I explained you about this. At first, enzyme bind to substrate. And during an reversible reaction, it can convert to ES complex. And after that, we have E, enzyme plus product. That enzyme again regenerate and product go for its work. And these are the application of the things that I bring here just for your better understanding. Guys, I want to know about Michaelis-Menten kinetics. Guys, you should memorize this formula for your final exam. Okay? This is the maximum velocity. This is the initial velocity. This is substrate concentration. And this is the Km or constant of Michaelis-Menten.
Half of the matrix game, right?
Yeah, exactly. Exactly. And this is the thing that are written for better understanding. And guys, do you know what? This Km reveals the affinity of enzyme to substrate. As the Kmb is small, it has high affinity of enzyme for substrate. And if we have larger Km, the affinity of enzyme for substrate is lower. Okay? It is inverse somehow. The small Km, high affinity of enzyme for substrate. The large Km, low affinity of enzymes for substrate. It is better, no better, necessary to memorize all of these parts of the system for your final exam. Have a big green tick above that. Okay?
Ma'am, I have a question. What's the difference between normal enzyme and an allosteric enzyme?
Allosteric enzymes have different subunits. Okay? And we can't actually discuss and explain the function of that based on Michaelis-Menten equation. It's not working for allosteric enzymes because they have different subunits and they are working cooperatively. Exactly like hemoglobin that bind to oxygen. When the first subunit attached to oxygen, another subunit attached to oxygen is easier than the first. And this is a cooperativity effect because of conformational changes. It is, again, the same for allosteric enzymes. Okay?
Can an allosteric enzyme bind to active site or does it only bind to allosteric site? You can
Allosteric, okay, it's called allosteric because it binds to another place than the active site. It cannot bind to the active site.
sometimes it
We
binds.
say allosteric.
For now, we have just reached the level that it only binds to it. If you want to overcomplicate yourself, then you can do that.
I will explain again. Allosteric is, for example, we say that these enzymes have some effector allosterics. Okay, effectors. They allow means another. That means, for example, this is your enzyme for 3 subunits. These are active sites. We have some effectors that attach another part except active site. I will explain you because we will discuss about homotropic and heterotropic allosteric effectors. Right, about the Michaelis-Menten kinetics, you should know about the Michaelis constant Km. That our friend explained about the Km, that it is the affinity of the enzyme for the substrate. Could you repeat the definition of Km again?
It is half of Vmax.
Km doesn't vary with enzyme concentration. It is a constant that it is just for this enzyme. The Km for this enzyme is constant. Guys, I've explained you about high affinity and low affinity, and let me show on the curve. Here you can see the reaction velocity, and here is substrate concentration. And we are speaking just about general enzymes, not allosteric enzymes. Okay? About general enzymes. You can see that here is half of our Vmax. Am I right?
Yes.
This enzyme with blue curve have lower Km, and the other enzymes with red curve have higher Km. What does it mean? The higher Km, the lower velocity. Why? Because the affinity. It shows the affinity of enzyme. Because here the affinity of enzyme 1 to substrate is high, we can see that in lower substrate we will reach the half of Vmax. But in this situation, we need more substrate to reach higher half of Vmax. And this is why the higher Km is lower affinity. Did you get that point? No? No, Nitya?
It's important for the example.
The higher affinity, the lower affinity, that's more important. Just
And about
memorizing it, that's
that Km, higher Km is mean lower affinity.
lower.
Yeah, and lower Km is higher affinity. Did you get all of you, guys? Okay. Guys, about another thing, I want to tell you about reaction order. Do you know about that? Yeah.
First order
First order, zero order. Zero order and first order, exactly. Do you know?
Yes.
I want to explain a little for all of you guys. First, when the substrate concentration be greater less than Km, okay? Be less than your Km, your reaction will be second order. What does I mean? I mean that. What do I mean? I mean that when your substrate concentration be lower than Km, the enzyme reaction velocity be first order. With increasing substrate, the velocity will increase in a linear shape. With increasing the substrate concentration, the velocity will increase in this form. It is first order reaction. Okay? But after that, if the substrate concentration be higher than Km, this is zero order reaction. Why? Because the enzyme active site is saturated. Because in our body, normally, in our body, the concentration of substrate is lower than Km. And that's why our enzymes are working efficiently. In a first order reaction, they won't reach in this area because the substrate concentration is limited. Okay, this is the physiology of our body. Guys, I want to tell you about a matter of the Lineweaver-Berg plot. Do you know what is it? No, but it is completely related to Michaelis-Menten equation, and it's not new things, but it is for having better the effect of inhibitors on and the study of the enzyme. Guys, do you know these studies that I'm telling you about the velocity and the Km, they just separated the enzyme enzymes from the cells, and these studies occur in the test tube, not in our body. These are in vitro, not in vivo. Do you know about the meaning of in vitro and in vivo? All of you?
Yes.
Yeah. In vitro means in test tube, in laboratory situation. For example, for my PhD thesis, I was working on breast cancer. Not breast cancer tissue, I was working on breast cancer cell lines. This was an in vitro study. We have a media that just I harvest the breast cancer cells here. This was an in vitro study. But if you try to do the work in animal models or in human models, these are in vivo studies. In the live creatures. Animals or humans. Animals, we call that experimental studies, but there is no difference. They are in vivo studies. Like, for live Weber plot, because here you can see the Michaelis mental, okay? You can see because of the hyperbolic nature of the curve, the evaluation of velocity is not easy. They try to convert it to a linear situation. What did they do? They inverse the equation. Is there any volunteer to come here and inverse this equation completely? Why? It is so easy.
I dropped math for the reason. I reached math.
I will choose one of you and I will help you. Just you should reverse the equation in this form. Is there anyone?
Vmax equal K.
Thank you.
Do I have to solve this equation or
Just write the final exercise. Yeah, completely. Like a piece of cake or piece of pizza. Good job. Omit this and tell me. If I wanna write, do you know about this equation? Yes. If you wanna tell me which of them is Y, which of them is A, good job. Clap for her. Okay, guys, I want to tell you, if I want to say you and draw the equation till now, this could be as y. This is our x. This is our slope, and this is our b. You can see here the equation completely here, and if I want to draw the curve, you can yeah, you can see here that we have 1 over the V max as intercept here and minus 1 over Km as intercept here. Guys, you can do it. If you want, we can do it here, but I'm just a little worried about the time, but we have enough time. But we can, at first, imagine that our 1 over V0 is 0 and assess this intercept, and in other situation we imagine this is 0 and evaluate this intercept. If you